Okay, this is how DNA was cloned, and most of this kind of work was carried out in the sort of 80s, and it was the way in which the sequence of bases for a lot of genes were determined. And if you get the sequence of bases, then you can infer from that the amino acid sequence. So there's an awful lot of useful information came from this approach. Right, so let's say if you wanted to clone the gene for hemoglobin, right? So hemoglobin, of course, is it's made up of two proteins, made up of the alpha chain and the beta chain. So let's say you wanted to find out clone the genes for those. So what you'd have to do is you'd have to we want to find a cell that's making a lot of mRNA for those genes. Okay, and the reason why we need the mRNA is because we're going to make a DNA copy of that mRNA, and then clone that piece of DNA. So you have to choose a tissue which is going to have a lot of hemoglobin mRNA in it. Most cells are not making any hemoglobin in them. The only ones you are the red cell precursors. Okay, so of course red cells themselves don't have any DNA in them at all because they haven't got any there's no nucleus, there's no protein synthesis going on in there, but the precursors are found in the bone marrow, and that's where they're making loads of mRNA for hemoglobin, making loads of hemoglobin protein. If you wanted to, on the other hand, if you wanted to clone the gene for insulin, then you probably want to get a bit of pancreas tissue, because that's where insulin is. The beta cells of the pancreas are making loads of insulin, so they'll be making a lot of mRNA. Okay, so depending on which genes you're really interested in, you choose your tissue accordingly. And then what you've got to do then is extract the mRNA. From the cells, which isn't too difficult. We won't go into the details of that, but just to remember that mRNA is, it's the reason why we're going for mRNA is because it's fairly simple, because when you first make the mRNA, they're the coding regions, and these red bits are the introns, and they're non-coding. And what happens in the processing of the RNA, of course, is those introns are spliced out. So, sorry, you will end up with and those red bits, the introns, they get chopped out, they kind of looped out, and then the ends are joined together. So we end up with a piece of RNA without any non-coding regions in it. All codes for protein. There's a bit on the end which is a poly A tail and stuff, but that's not coding, but we won't worry about that too much. So that's why it was simpler to go for mRNA, because you've got all those non-coding regions are all gone. It makes the piece of DNA much smaller and easier to deal with. Okay. Also, you know, certain cells are making lots of that mRNA, so the chances are you're going to be able to get hold of a copy of that gene. Right, so the first job that has to be done is you have to get the mRNA, which I'll draw in blue. So there's a piece of mRNA, and that is used, you had an enzyme called reverse transcriptase. And that does what it sounds like it does. Reverse transcription is mRNA into DNA. Well, what this does is it uses the mRNA as a template, and it makes a copy of DNA. So that's a complementary sequence of DNA. That's why it's called copy DNA, cDNA. Okay, so it matches the base pair sequence on the mRNA. As I say, normally in transcription, what happens is DNA gets converted into RNA. It's the exact opposite. The enzyme reverse transcriptase is found in viruses, which have an RNA genome. So that's where it's purified from. So you've got your cDNA. Add an enzyme to dissolve all the an RNase, which will dissolve all the RNA. So you'll end up with single-stranded cDNA. Okay, so all that RNA is gone. And then you have to convert that into double-stranded DNA by adding DNA polymerase. So you've got double-stranded DNA. And that will be a piece of DNA which will give you the gene sequence for the mRNA it came from. Hopefully, if you're doing hemoglobin, it might be the alpha, one of the alpha chains, or it might be the alpha chain or the beta chain. So that's single-stranded there, and that's double-stranded DNA. Now, that DNA, before you can do anything with it, you have to give it these things called sticky ends. Okay, and I'll explain what these are. Right, so in cloning, you use enzymes called restriction enzymes. And these cut DNA. They cut it at specific sites. There are several hundred of these restriction enzymes. They all recognize different sites. And the one I'm going to talk about is a very commonly used one. It's called BAMH1. And that's isolated from microorganism. Bacteria have restriction enzymes in them to cut up bits of DNA which are introduced into them by viruses which infect them. So it's like an immunity for the bacteria, but they're being purified and they're now used as tools. And BAMH1 will recognize this site. It recognizes whenever it sees GG, AT, CC. Now that's a 5' end and that's a 3' end. Now let's look at if it's double-stranded, that's going to be CCTAGG. Now you can see that site is the same whether you read it backwards or forwards. It's got symmetry. It's called a palindrome. And all restriction enzymes, they all recognize palindromic sites. They've all got this symmetry. What BAMH1 does, it will cut the DNA there and there like that. So when it's cut the bit of DNA, if I draw it, I'll show you what it will look like. So I won't do all the sequence, but it's going to be then that final G. And here, on the other side, it will be C, C, T, A, G. So it's longer on the bottom edge than the top edge, and of course, when it cuts the other one, that's going to have a corresponding sequence of G, A, T, C, C, and G there. And you can see that these two, if they move together, they can rejoin. Okay, so if you cut one bit of DNA with the same restriction, 2 bits of DNA with the same restriction enzyme, you end up with one sticky end there and another sticky end on the other bit of DNA, and then you can slot the two together. And that's what we need to do with this piece of double-stranded DNA. We need to make it have sticky ends. So let's draw that down here. So we've got our double-stranded DNA, and we add short little bits of DNA. So usually 10 or 12 amino acids, 10 or 12 bases long, to give it sticky ends. So it's got that bit there, and it's got this corresponding the other sticky end, the sort of mirror image of it on the other side. So these, you've added the sticky ends, so you add short sequences of nucleotides. And to join those green bits, the short sequences, onto the main cDNA bit, the red bit, you need an enzyme to do that. It's called DNA ligase. DNA ligase can join together those two blunt ends. Okay, so you've now got that, and it's all ready to stick into this bit of DNA here. The clone bit of DNA is all ready to stick into a plasmid. Okay, that's going to go into a plasmid. Let's remind ourselves what a plasmid is. A plasmid, right, so you have bacteria like E. coli. We're going to stick this into E. coli. And you'll have the big bacterial chromosome, but also you have other bits of DNA, little circular bits, circular double-stranded small bits, and they're called plasmids. And they can actually, bacteria are a bit leaky, and those plasmids can pass out of one bacteria into another one and transfer bits of DNA from one bacteria to another. And that's in fact how antibiotic resistance genes are spread. So it's obviously, in that case, it's not a good thing, but it's very useful to use in this cloning technology. So the plasmid we get is a very commonly used one, and it grows in E. coli. It's called PBR322, and it's a circular bit of DNA. It's about 5,000 base pairs long. I think P stands for plasmid, BR stands for someone's initials, the person who made it out of other bits of naturally occurring plasmids. And it's useful because it's got a gene there for tetracycline resistance. So tetracycline is an antibody. And it's got a gene here for ampicillin resistance, and that's going to be very useful, as we'll see in a minute. Ampicillin resistance. And it's got a sequence, the six nucleotide sequence there, right in the middle of the tetracycline resistance gene, which is recognized by the restriction enzyme BAMH1. So the next thing to do, we've got to remember what we want to end up doing. We want to end up sticking that bit of DNA into the plasmid, and it's got the right sticky ends to go into this site here, the BAMH1 site. So right, so you get a load of this plasmid. Where do you get the plasmid from? You purify it from E. coli. You grow E. coli, which has got it in it. You purify the plasmid. So here's a test tube full of plasmid. In there, you're going to put in BAMH1 to cut it. And then you're going to put in your cDNA with the BAMH1 sticky ends. And hopefully what you're going to end up with is some plasmids which are like this. So we've still got our ampicillin gene, which is intact. We've still got bits of the tetracycline gene. This is no longer, because what we've done is, well, there's the BAM site there, the sticky end there, sticky end there. I'll draw the new bit. This is the cDNA. I'll draw that in yellow. That's the insert. That's the cDNA insert there. Right, so that is now, we've got a new bit of DNA in that plasmid. Hopefully in this test tube here, we're going to end up with some of that stuff. Inevitably, we are going to end up with some of those plasmids which haven't been cut by the enzyme perhaps, or perhaps they do get cut and they just join back up. Two sticky ends here just join straight back together again. And so that's going to be no good, because that will not have our cDNA in it that we want. So what do we do with this tube of fresh plasmids is we're going to mix it now into a tube of E. coli. Okay, and you can make those E. coli a little bit leaky, so they're better at taking up the plasmid. So the contents of that tube is going to go into there, and hopefully you're going to end up with some. Here's an E. coli cell. There's its chromosome. Hopefully you are going to end up with some that have got the plasmid. That's supposed to be the plasmid, and that yellow bit is supposed to be the new bit of DNA. And there'll be multiple, well, maybe just one copy of that. So that what we're going to do then is like this, get this E. coli, and we need to find out. We'll have millions of E. coli in here. Lots of them are going to be no good because they'll have no plasmid at all. They're no good. We don't want to grow those up. Some will have plasmid with no insert. We don't want them. They're no good. Hopefully we'll have some plasmid with this cDNA insert. So the job is now to try and screen them. It's called screening. We need to find out which ones have got the insert, and then we're going to keep them, and then we can make that E. coli grow and grow millions and millions of versions of it, and we get millions of copies of our inserted DNA, and then we can sequence it or whatever that's going to do with it. All right, so let's look at the screening process. So you get the E. coli and you plate it out onto a Petri dish, an agar dish, all right? And on this dish, we're going to have the antibiotic ampicillin. Just call it AMP. So it's agar with ampicillin. So that means ones with no plasmid, yeah, they won't grow, because they need the plasmid in it to make it resistant to ampicillin. So we're going to end up with like lots of spots on there. So you dilute out your E. coli, and you'll end up with maybe several hundred little spots. Each one of those is a colony of bacteria which have all derived from one. So you put a little bit of liquid on that plate, a drop of liquid of your E. coli solution. You spread it around. You get it the right dilution so you don't end up with like millions and millions, because you wouldn't be able to, they'd all run into each other. So you just want a few hundred on the plate. And each one of those has taken up the plasmid. Now we need to find out which of those ones has got the plasmid with the insert. Now how are we going to do that? Now if you remember, here's just to draw a plasmid here. Again, right, so we've got the tetracycline insert there. Sorry, the ampicillin insert there. But here we have the tetracycline gene, and that's in red there. But that's not working anymore because it's been chopped in half, and we've got our yellow cDNA insert. So the colonies here that we're interested in are the ones which are not resistant to tetracycline. So I'm just going to simplify this by, instead of having all those, I'm going to draw 5. Just draw 5. So here we end up with 5 colonies, like in the shape of on a dice, yeah. And what you do is you take a block. It's called a replica plate, yeah. So that is blotted with a piece of blotting material, transferred to another plate. Here's another plate, but this one contains the antibiotic tetracycline. So let's say that only that colony and that colony grow. That means that these 3 colonies here, that one, and that one, they don't have a tetracycline gene. So these are the ones with the insert. They're the ones we're interested in. So then you pick off those colonies one by one. I've only got 3 of them there, and so I'll draw 3 little flasks. You maybe end up with an awful lot more than that. And you put that one into there, that one into there, that one into there, and you grow the bacteria. So you'll end up with millions and millions and millions of those bacteria, each of which will contain, which has got the the plasmid with the insert. And that's why it's called a library. So you end up here with a library, maybe several hundred different bacteria. and they all contain some kind of insert. Now, if we're interested in the say the alpha hemoglobin gene, then we're going to hope that one of them, the insert, was made from a piece of alpha hemoglobin DNA, and there's a good chance of that if you've chosen, you know, a cell which is making loads and loads of alpha hemoglobin mRNA. So you grow that up, each one, and you investigate it, right? So you grow it up, and then you've got to extract the plasmid DNA. So you chuck away most of the DNA. You lyse the bacteria. You get the DNA, and you can separate the plasmid DNA. It's got a slightly different density to the chromosomal DNA, and that's what you're interested in. And of course, then we need to chop out our bit of DNA we're interested in. So you're going to end up with, here's the plasmid here. Yeah. Okay, so I'm going to copy that. Let's move it down a bit. So we want to get the bit of DNA of interest out of there, so that should be there. So how do we do that? Well, we want to chop out that yellow bit of DNA. How do we do that? Well, we know we've got a BAM-H1 restriction site there and there. We cut it with the BAM-H1. And you're going to end up with your bit of DNA that you're interested in there. And I've only drawn one plasmid there. Of course, you're going to have potentially billions of these pieces of plasmid. So you've got billions of bits of DNA you're going to have a lot to work with. And you can use this, so then you can, this could be used for sequencing, it could be sequenced. And you could, by looking at the sequence, you can tell, is it for the alpha hemoglobin gene? Right, so you may think, well, you know, someone had to, how did you know the sequence of the alpha hemoglobin gene first? Well, that would have been done by things like, you would have, first of all, you get the alpha hemoglobin protein. So proteins were, and then they can be sequencing amino acid. And from that, you can infer some of the nucleotide sequence. And usually you wouldn't have to sequence the entire protein. You could just do the first, say, 10 amino acids. And so that would give you the first 10 amino acids would give you, the first 30 nucleotides, yeah. And you could just look for that 30 nucleotide sequence, which is a lot easier than looking for the entire gene. So that's an early approach to how multiple copies of bits of DNA were obtained by using a cDNA library.